Thank you very much. First, I'd like to begin by thanking the AACR for the opportunity to share our research as part of its press program. So today, I'm going to be presenting our work on DNA methylation to predict tissue of origin in cancers of unknown primary, or CUP. Our work is going to focus on developing, or this work focuses on developing a robust and biologically grounded classifier using a reduced set of CpG markers. And this is my disclosure slide. So, CUP represents metastatic cancers where the primary site cannot be identified. This makes treatment difficult, and while a minority of patients are able to receive site-directed therapy, the vast majority receive empiric chemotherapy, which results in poor outcomes. This highlights a need for improved tools to help identify tissue of origin. DNA methylation provides a stable tissue-specific signature that persists even in metastatic tumors. This makes it a promising approach for identifying tumor of origin, potentially being more robust than other molecular methods. Our goal here was to develop a classifier that can predict tissue of origin using a focused set of CpG sites rather than hundreds of thousands, which most of the panels now comprise. So we want to basically improve practicality while maintaining strong performance. For this study, we analyzed over 7,400 tumors across 21 cancer types, and these were from the TCGA dataset. And so basically, using our feature selection model, we reduced the number of features from 450,000 to about 1,000. From here, we trained and evaluated our model, and using stratified splits and independent validation sets, we were able to further evaluate these. So one other thing that I want to highlight here, that we use also cross-platform validation using some of the more current platforms. Then after, we also assessed the biological relevance of these selected CpG sites. Here, one thing that we can importantly is we see that the selected CpGs, or preserved cancer type structure, looking at unsupervised clustering, we can see that we have clear separation by cancer type, and these are in agreement between the phenotypic cluster assignments and their tumor labels. Moreover, these clusters were able to clusters were mostly pure, corresponding to the single cancer type. And these, however, some of these showed some overlap, particularly with some biologically similar tumors, such as colorectal subtypes and gynecological cancers. So what this suggested for us is that these features also capture the underlying cancer biology. We also found that the methylation patterns were consistent with gene expression profiles. As shown here, gene expression profile also preserved the cancer type structure, and methylation was inversely associated with gene expression, which was what we would expect. And this provides and gives a little more evidence that this model reflects the underlying tumor biology rather than just looking at statistical patterns. And looking at model performance, we were able to see high F1 performance with our test set reaching about 0.945. When looking at independent validation, we were able to see good performance. However, we did see some misclassifications, and these were usually associated between biologically similar cancers. For example, colorectal cancers being classified as rectal adenocarcinomas being classified as colorectal cancers. So when we combined these subtypes, we were able to improve accuracy. And with our expanded cohort, we were able to maintain good overall performance in the model, reaching almost 0.9 in the F1 score. Another thing that we wanted to explore was whether the performance of our model was influenced by factors such as class size, heterogeneity, or tumor purity. And here we did not see that any significant association with these data set characteristics. So instead, what this means is that performance differences are likely to be associated with intrinsic biological variation across the different cancer types. So just to summarize here, we developed a reduced CPG panel that can robustly predict tissue of origin site across different data sets and platforms. We were able to capture meaningful tumor biology with this set, and classification challenges that we saw were mainly reflect biological complexity rather than technical limitations. Together, these findings support the practical development of this tissue of origin classifier for CUP classification. Moving forward, we want to investigate this model in actual patients, true CUP patient cohorts. We're also looking and exploring into moving this into liquid biopsy, as it could enable maybe less invasive testing. And ultimately, our ultimate goal is here to support a more informed and personalized treatment for decision making. And then just lastly here, I'd like to thank our team for all their hard work, especially Dr. Kazuto Nisha, who's the study PI, and Dr. Kazuko Sakai, who's the co-lead on this project. Thank you very much.
Thank you so much for the great presentation. I believe that cancers of unknown primary are definitely one of the more challenging problems in oncology, because we don't know their site of origin and their eligibility for clinical trials and their treatments. It's so challenging for oncologists. And so even now that we have molecular profilings and there are some advances, still we are not able to predict the cancer type of origin. And integrating this would be very useful, like really having a fingerprint of the tumor and really trying to predict what is the origin and direct the treatments, especially early on, because the choices of treatments matter for that patient. And so let me ask you a first question, like how do you see this in comparison with just the regular DNA NGS testing, and how this would complement that?
Well, yeah, and yeah, this is a very important point, because this is not meant to replace the key oncogenic drivers. This is supposed more to complement and basically provide answers for those cancers that we don't have any answers to. Yes. And so, and another thing that I think actually we're going to be looking for and doing more profiling, because what we actually are also identifying is phenotypes. And then we can more interrogate these phenotypes. Are there any possible druggable targets within this phenotype? So then we are not looking necessarily for just the tissue or the origin is, but what phenotypes does the present tumor actually may have? And can that help inform to guide the therapy?
I see. Let's open up for questions from the room. Any questions? Yes.
Hello, Matthew Herper from STAT. I'm just struck that the existing multi-cancer detection test from GRAIL is a methylation-based test that locates site of origin, because obviously with the multi-cancer liquid biopsy you don't know, and they had to figure out a way to do that. I'm wondering how the research converges and how your methylation work is similar or different from that and what we know, because it seems like there might be a use there that is maybe not their intended use for the test, but might actually be helpful.
Right, and yeah, so I mean, this is methylation.
And also I'd love to know a little bit about differences in how much sequencing and depth are needed between what you're considering doing and what they do, which I imagine is very expensive, what they do.
Right, and so there's a couple different layers here that we have to look at, and DNA methylation promises to be a better approach because you can maintain characteristics from the tumors that we're using to build these models and their tissue of origin. And so when we're talking about cancers of unknown primary, we're already talking about advanced cancers that have already metastasized and have already evolved and changed quite differently from what would have been their primary. So what we're trying to capture here is basically the origins. And this, I think, one of the reasons why many other approaches like transcriptomic profiling has failed. Now, with regards to how we are looking and how we're, developing the different tests using methylation profiling, like you mentioned, it just, I guess, depends what questions we're looking at in our to identify these markers. So when we look at the features, we can identify for just strict regression classification, or are we looking for also some biological meaning? And I wanted to incorporate that in our model to where we can actually find interpretable sets of CPGs that can help us also inform, not just for strict classification, but also inform us biologically. And as far as how deep, I mean, that's something that we're still working on. We don't know the precise answer. This is the first working model, so we're going to be testing and going a little bit deeper. So I'm sorry I don't have that information right now. My other colleague is the one that's really the expert in that part. So,
Any other questions?
I have two questions from reporters on Zoom.
Yes.
The first is from Lisa Astor with ASCO AI on Oncology. She asks, can you elaborate more on the machine learning techniques used to train your model?
Sure. So for the feature classification, we actually used gradient boosting. And then for the prediction model, we used logistic regression. And those details are explained more in our poster presentation. I can go over those in more detail there.
Thank you. The next question is from Lynn Peterson with Trends in Medicine. She asks, how far away is this from general use? What are the biggest challenges to getting this into general clinical use?
Yes, well, the first thing that we have to do is, like I said, we have to test this in actual CUP patients, and actually that's where we're at right now. We're going to start working up setting up the protocols. Our clinicians are working on this to set up this. Get the patients and set up the trial to evaluate this. So, I mean, I think our model is fairly ready to be tested in actual patients, and we'll just see how it goes from there. So hopefully within the next year or so we can start testing. And if it works well, we can see how that progresses and moves forward.
And just as a follow-up question from that, so do you think that when there is a diagnosis of a patient with unknown primary, we can request this, and what would be the turnaround time and how much tissue you need?
Yes, and so that's a good question. And one of the things why we want to focus on this, use a focused set of CPG is to reduce the cost, reduce the time, and enhance the processing. So I don't know exactly what would be the cost because, well, we're working this in Japan, so
yeah.
in the U.S., but it would be much, much cheaper than using the current methods. I mean, the current versions of Infinium, now you're looking at close to 1 million different CPG sites. So the amount of tissue that you need and the time and to analyze that, it's longer. So hopefully this would be a much shorter process. The specifics I'm not sure right now, I can tell you.
And are you working on any blood-based?
Yes, actually that's the next thing because obviously not every patient can get tissue. So the next challenge is, can we use circulating DNA for this, and how similar or different would that be? Would our model work on that? And that is what we are working on right now at the present time.
Oh, wonderful.